So this sample bag means the sample is removed, put into the one bowl, and root and soil separate the root and soil. Now we should one thing kept in our mind, which type of nematodes present in roots and which type of nematodes present in the soil. This is very important things. Before extracting the nematodes, we should know that if we are extracting nematode from soil, so which type of nematode and which stages of this nematode we can get by the method of the extraction of soil, or if we are talking about the roots, so which type of nematodes and which type of the nematode stages we can isolate it by the root system. In our the classification of nematode according to their feeding habit, in this we already discussed that nematodes are categorized into the three important category, that is endoparasite, semi-endoparasite and ectoparasite. And these three categories further divided into two categories, that is sedentary nematodes and the migratory nematodes. So in the soil and in the root, different types of nematodes are present. Okay, like in roots, so semi-endoparasitic nematodes are attached with the root, endoparasitic also attached with the root. The root system. The sedentary of the parasitic nematodes are with the root, but the procedure is different for extracting of different types of nematodes. Now, first we so today we will extract the nematode from the soil. Okay, so by the soil, which type of nematode we can extract? So in this soil, all saprophytic nematode, free living nematode, soil dwelling mononchytes, they are present in soil. They are not present in the root system. Thereafter, some of the free living stages of the plant parasitic nematode, ectoparasite nematodes like criconematids, dorylamids, they are present in the soil. Okay. Thereafter, some of the stages of semi-endoparasitic nematode or endoparasitic nematode like the root knot nematode juveniles are present in the soil. Means after the hatching of eggs, the juveniles comes into the soil and they are searching towards their host. So they are also present in soil. The male of root knot nematode also present in soil. Male of heteroid nematodes are also present in soil. The juveniles of the different kinds of plant, means all infective juveniles are present in soil, and the especially like some of the semi-parasitic migratory semi like hop Thailand sedentary, they are migratory semi-endoparasitic nematodes and they are present in soil. So in this soil, the all kinds of nematodes are there. So how to extract these nematodes? So today we discuss the three important methods. There are so many methods for extracting nematode from the soil. You can also develop one method according to your availability and suitability. So today we discuss the three important methods on the different three principles. Like first we discuss the Beerman funnel technique, which was first time reported by the Beerman in 1970, in which Beerman took one the funnel attached with a rubber tube. You can see here the rubber tube, funnel attached with the rubber tube and fix it with the clamp. This whole is filled with and then soil, water and put it in funnel. This was done by the Beerman in 1970. What will happen in the cotton bag? The nematodes are there, after getting the moisture and the water, the nematodes move towards the water, so that they are comes out from the cotton bag, and thereafter they are settled down at the bottom of the rubber tube. Next day you just open the clamp and collect the suspension of nematodes. Thereafter this cotton bag will replaced by the aluminum wire gauze and a tissue paper. In this method, the aluminum wire gauze, put it on the funnel, and then put it on the wire, and then the soil, put it on the particular paper. This whole assembly is already filled with the water. So this is the method of Bierman funnel technique. Now we are also using this method, and this is very easy method. The merit and demerit we will discuss later after discussing all three methods. So now the whole assembly is replaced by a tray. That's why it is called as modified tray method. Means this whole assembly is replaced by the tray. Means you just put the wire mesh or tissue paper on the tray. So this is the modified tray method. Okay. Accordingly, for tray, the size of tray is very large, so that we have to put a large sized aluminum wire gauze. So now we put the aluminum wire gauze, and thereafter put a tissue paper, okay, and then put a soil. But when you extract the nematode, you should know how much soil you have taken. Like in Indian condition, we are taking 200 cc soil. 200 cc soil. It is the ideal sample size in India. Now I am taking 100 ml beaker. So I am taking 200 cc soil, means 1. Thereafter 2. Take. Now this soil is 200 cc, means cubic centimeter. Thereafter, from one side, this tray will fill with the water. Thereafter, cover it and place it. Cover it and place it for 24 hours. Just a minute. You can able to see or not? Yes, no problem. So next day, after 24 hours, what will happen? Nematode will penetrate the tissue paper, and they all are settled down at the bottom of this tray. Now all the nematodes which was present in the soil, they all are comes down or comes into the water and in this suspension. Now you can calculate, you can count this nematode, you can identify the nematode, whatever present in the 200 cc soil. So this is the suspension, and you can collect it into a beaker. So this is the method of extraction of nematode. by the modified tray method or the Bierman funnel technique. In this method, only the active nematode can comes out from the tissue paper, and then you can collect into the by this method. The second important method, which was given by Cobb in 1918, in which again you have to take 200 cc soil into the in the bowl, then mix with the water. Because this method is decanting and sieving technique, so the soil is mixed into the water. Wait for 15 seconds and then you just take a different size of sieves. We will start with the 60 mesh sieve, thereafter 100 mesh sieve, thereafter 200 mesh sieve, thereafter 400 mesh sieve. Means different type or different size of sieves we are taking and sieve out the particular suspension. So different types of nematodes will collect on the different size of the sieves. Means we can collect the nematode according to their size. So according to their size you can collect the nematodes, and we can also collect all kinds of nematodes present in this soil. You can collect by these different types of sieves. The size of sieves you can read on the on the sieve. This is the 400 mesh sieve. This is 200 mesh sieve. Likewise, this is 60 mesh sieve. So you should start with the 60 mesh sieves. So I am not taking much time, but I just show you the procedure to how to extract the nematode. So first stir it and wait it for 1 minute so that all nematode or soil particle will settle down. But the whole heavy particle of soil will settle down. Nematodes are moving or swimming into the soil, and thereafter you just sieve out by the sieve. So decanting and sieving technique, you can see the soil. So the heavy particle of soil will be settled down, and now this is the 60 mesh sieve. So you can only collect the cyst nematodes and very large size nematode. Otherwise the other nematodes are present in this suspension. Now you just further decant or sieve it by the different size of sieves so that you can collect the nematode in size of the sieves according to their different size of nematodes. Means different size of nematode collected on the different size of sieves. Means the numbers will increase, then size will decrease. So on the 400 mesh sieve we can collect all kind of nematodes or juveniles also. So this is the method of the Cobb sieving and decanting technique. Now we will discuss the third method, and it is given by Fenwick, a scientist, and give the assembly that is called as Fenwick cane assembly. In this you can see the cane. There is one cane. You can see the design of this cane. This is the simple method in which you just put a one 16 mesh sieve on the the 16 mesh sieve on the top, so that the root debris and heavy particle of soil will be separated on the uppermost 16 mesh sieve, and nematode will comes out in the bottom of nematode and soil. All will come out in the particularly bottom, and like that you just see this is the soil. Put it on the top of the sink so that it will be more easy. And this technique is required running tape water. And this method is very useful for the extraction of a cyst nematode. You just put a sieve so that water will float from the collar with nematodes, because nematodes are floating into this particular cave. The heavy particle will be settled down at the bottom, and nematodes are floating and swimming into the water, and they will by the flow of water they will collected by this help of the collar. Like that you just see. I need some more water. So in this method the nematodes are collected by the flow of flotation. Here you can see we are taking one sieve here, so that and this technique is put it near at below the tape, so that the running tape is continuously so this is the third method, that is the Fenwick cane flotation technique. And in this method the nematode will collect by the flotation technique. So whatever the nematodes comes out from this method, they will collect on the sieves, and thereafter you can collect these nematodes by this sieve. So thank you very much. Now we will discuss the manual technique. We can extract all active nematodes only. We cannot extract the inactive stages. We cannot extract the eggs. We cannot extract cyst nematode by Bierman funnel or modified tray method. Okay, the active nematodes can comes out by this method. Second thing, like the very sluggish nematode, that is the criconematode, hemicyclophora, due to their scales and spines, they need more time to come out by this method. They need more than 24 hours. That's why the nematodes cannot prefer if we extract these nematodes. So this method is not suitable, means modified tray method is not suitable for extraction of this method. Benefit of this method is modified tray method. Benefit of this particular method is by in this method the water is not much required. The equipment is not more required. We need two plastic tray, one half 1 feet by 1 feet, the aluminum wire gauze, and need one tissue paper. So this is very easiest method, not required any electricity, not required any the skilled person. It is very easy and cheap method. So you can also use this method at your home also. So for extraction of active nematodes. But there is a demerit, because you cannot collect all kinds of nematodes. Okay. The second method, that is crop sieving and decanting technique. In this method you need this brass or the sieves, different types of sieves, means near about 1 to 5 sieves, like 60 mass sieve, thereafter 100 mass sieve, 200 mass sieve, 400 or 325 sieves. So you have to purchase a set of sieves like near about 5 to 10,000 rupees required for the extraction of nematodes. Thereafter you need a tape water, means in this method for extraction of nematode from 200 cc soil, at least you required 5 to 10 to 15 liter water, but it will increase if the soil is clay. So according to the type of soil, if sandy soil is there, so 10 liter is sufficient for extraction of the nematode. But if clay soil is there, so maybe you require 20 to 25 liter water for extraction of nematode, and the skilled person is required for extraction of nematode in this method. And in this method you can extract the nematode near about 15 to 25 minutes according to skill of this person. So near about means we can say that yes, half an hour is required for extraction of nematode by cob sieving and decanting technique. But in case of Beerman funnel technique, you should wait for 24 hours. You should wait for 24 hours. Means if you are putting soil sample today, so the soil nematode will extract next day. Okay. So this is the limitation of this particular method. But in this method, the water is more required. Thereafter, the nematodes, if you are having a nematode extraction lab, or your drainage water should also manage, otherwise nematode will disperse by your drainage water, because if you are using this method, so nematode will also running through the drainage water. Thereafter, the third method is the flotation technique, and this method is also very good for cyst nematode, because cysts are floating on the water. That's why it is a very good method for extraction of cyst nematode. But we can also collect all different types of nematodes by this method. But in this method, we cannot collect nematode separately. Means like in cob sieving decanting technique, you can extract the nematode according to their size, but in this method you cannot collect the nematode according to their size. All kinds of nematode you have to collect on a single sieve, and this method is also required more amount of water, means the running tape water required for extraction of this method. If you are talking about cyst nematode, so this method is near about 5 to 10 liter is required for extraction of cyst nematode from one sample, and depending on the soil. If you are talking about the other nematodes for extraction of the different kind of nematode by this method, so it required more amount of water, but we are using this method for extraction of cyst only. Okay, so thank you very much. Today